So we've talked about the cortical structures on the more detailed levels in this table, and now we'll describe the highest level of cortical physical organization, cortical areas. To begin to understand cortical areas, remember that if you look at the cortex from the outside, it looks a bit like this. And this is how the inner surface of one hemisphere appears, notionally looking outwards from the center. If these views are drawn in a simplified fashion, there are a few deep fissures or sulci that are prominent in most brains. This is the lateral fissure, the central sulcus, and on the inner surface, the cingulate sulcus. And as I mentioned before, the cortex is deeply folded, and there are lots of other sulci. And a significant proportion of brains have a generally similar pattern of sulci that looks a bit like this. The gyri between these sulci have also been named, the bulges, and the names are convenient for identifying different points on the cortex. Corbinian-Brodmann in the 19th and early 20th century divided up the cortex into areas, mainly based on differences in the cell size and cell density in the different cortical layers within an area. He identified about 50 areas and numbered them in the order in which he studied them. Now, since then, quite a few additional areas have been identified, generally subdividing Brodmann's areas, and I've illustrated a recent view of the areas relative to a few major landmarks. There's also been quite a lot of effort put into identifying the functions of different areas. But one thing to bear in mind is that the areas are not always located in exactly the same positions relative to the landmarks, which makes investigation of the functions of areas even more difficult. So, what distinguishes one area from another? The primary factor is the detailed arrangement of the layers. In detail, the pattern of layering can vary between areas, and one of the more extreme variations is seen in the primary visual cortex illustrated, which is the area Brodmann labelled 17, but is generally called area V1, and that's where sensory input derived from the eyes first enters the cortex via the thalamus. And this section is stained to reveal a high proportion of the neuron somas, and the usual absence of neurons in layer 1 is clear, but in this area there's a large layer 4 where inputs are received from the thalamus, and you can see a number of subdivisions in that layer 4, and these subdivisions aren't seen in the higher visual areas. For example, as I showed earlier, outputs from area V1 go to area V2, and in this picture you can see the area V1 and area V2, and the three sublayers of area 4 are visible in area V1, but at the boundary between areas V1 and V2, the sublayers merge to form a single layer. And in area V2, there's a simple, undivided layer 4. Another variation can be seen in the entorhinal cortex, where there's no layer 4 at all, and clusters of neurons called cell islands are visible in layer 2. Now, early on, cortical layering was classified into 5 types. But with more study, more subtle but significant differences have been identified, not just in neuron sizes, but in other factors. And this shows sections from 3 adjacent areas in the brain of a chimpanzee. And these reveal differences in neurons, in axons, and also in the concentration of a protein that provides structure to neurons. Differences between the areas are visible in all 3 types of sections. Within one area, the layering structure is the same for all these and some other factors, but there are significant changes at the boundary with another area. So this is how cortical areas are defined. Given this physical definition of areas, there's been a lot of effort put into identifying the functions of different areas, and there are some useful observations that can be made. The areas in blue are the visual processing areas, V1, V2, V3, and so on. The areas in lighter blue use visual inputs to extract information about the relative positions of different objects. And these areas make up what's called the dorsal pathway that we'll discuss shortly. The areas indicated in green make up what's called the ventral pathway that extracts information relevant to identifying the categories of visual objects. The somatosensory areas that process information derived from touch and muscle position are shown in pale green. Areas processing auditory inputs are shown in pink, and the red area on the border between auditory and visual processing plays a role in understanding speech. The motor areas that produce the outputs driving muscle movements are indicated in brown, and the yellow areas close to motor control for the head play a role in speech generation. The dark red area is called the frontal eye fields, and they play a role in directing the eyes towards objects that need attention. Information on smell is processed in the orange area. But there are then lots of other areas that are often regarded as playing a role in much less well-defined higher cognition. It's a lot harder to put any useful label on the functions of these areas.